Hi everyone, I am Verzola-Olivio and I am a PhD candidate at the University of São Paulo, Brazil. The results presented here are part of my PhD dissertation. In several species, the males exhibit courtship displays that can reflect their qualities and may be used by females to evaluate their mates and make their choices. These courtship displays can be composed of one or more signals from different sensorial modalities such as visual, chemical, seismic, or acoustic signals. Specifically, about the acoustic signals, its features usually vary due to sender attributes, like body size, dominance, and individual identity. Male guinea pigs and wild caves from genus Cavia exhibit a complex courtship display that's not common among mammals. Guinea pigs, Cavia porcellus, are polygynous and domesticated small rodents. Their social structure is based on the dominance relationship between males and long-term male-female social bonds. The males display a complex courtship behavioral sequence, which involves signals from different sensorial modalities, including a courtship call. The male initiates the courtship with contact behaviors directed to female. Eventually, the male can invert his perineal gland and mark the environment and the female. The contact behaviors intensify, and the male can initiate the rumba, a rhythmical movement of the male hindquarters, side by side. Whereas the male moves his hips, he utters the courtship call. Let's watch the display. The acoustic component of the display is a low-frequency and low-intensity broadband pulsed call, composed of brief and similar pulses arranged in a phrase, named PURR. Its pulse rate is positively correlated to the rumba. In the video, we cannot listen to the call, so I'll play it now. That's how the call sounds. Although several authors describe the acoustic structure of the PURR, there are no investigations about individual differences. So we aim to evaluate whether PURR is different between individuals. In addition, we aim to investigate whether it codes cues about male quality and whether it has the potential to influence the behavior of receivers, such as chooser females and possible opponents. We analyzed the acoustic structure of the PURR and hypothesized that it is individually distinct, and that the acoustic features reflect the male dominance status. We recorded 13 adult male guinea pigs during male-female pairing sessions, and 3 females were used to stimulate the males to exhibit the courtship. They were housed in a controlled room, and guinea pig food and water were offered ad libitum. The male and female were both placed in a wood-fenced experimental arena, where they were free to interact for 30 minutes. We paired each male 3 times, always with a different female. We recorded audio and video during the sessions. We only analyzed PURR phrases uttered during courtship episodes, so we identified them from the video records. A courtship episode is any female-directed contact behavior by the male, followed by a courtship display and mounting. The episode ended when the male stopped interacting with the female for a 10-second interval. We selected 10 PURR phrases of each male. One PURR phrase is a set of pulses with intervals of at least 0.1 seconds among them. We transformed the sampling frequency of audio records to 10 kHz to improve the frequency resolution. We measured 19 acoustic parameters, including spectral, temporal, amplitude, and energy features. We also extracted the frequencies corresponding to peaks of energy, possible formats, and the mean fundamental frequency using linear predictive coding. We determined the male dominance status using the individual dominance index modified David score. This index is based on the dyadic proportion of wins and losses of an individual. 8 males formed an isosexual group, and we recorded them daily for 1 hour in different moments between 8 a.m. and 6 p.m. for 15 days. We quantified the number of agonistic episodes and identified the winner and the loser. Agonistic episodes are aggressive interactions between two males and ended when one of the males retreated. The individual who retreats is the loser and the other one is the winner. Positive scores of the individual index are usually attributed to dominant individuals and negative scores to subordinate individuals. We used the coefficient of variation within and between individuals to access the potential for individual coding. Peak values higher than 1 indicate that between individual variability is higher than within individual variability, and the parameter has the potential to encode individual information. We used principal component analysis to investigate the contribution of acoustic parameters and explain the variability of the cause. We investigate the relationship between acoustic feature and the male dominance status using the Spearman correlation test. The modified David score represents the dominance status of each individual. We consider that acoustic parameters were strongly correlated to dominance status when the Spearman coefficient was greater than 0.7. Our results indicated that acoustic parameters of the PURR are individualized between males, but do not reflect males' dominance status. These results partly support our hypothesis, since we predicted that acoustic parameters are related to male dominance. Peak and PCA values suggested that the main differences between individuals are in frequency parameters. Peak values were higher than 1 for all the 21 acoustic features, and the highest values are minimal fundamental frequency, frequency 5%, and first quartile of frequency. The highest values suggested that these three parameters contributed more to individualization of PURR. The first principal component explained the most cumulative variance and was represented by spectral features such as fundamental frequency measures, quartiles of frequency, and peak frequency. Spearman correlation coefficient did not indicate a correlation between call features and the male dominance status. Individual differences in spectral parameters could be explained by differences in the vocal apparatus of the individuals, as predicted by the source filter theory. This theory states that spectral features of mammal vocal signals are a combination of two independent mechanisms, namely the source and the filter. Source features have their origin in the vibration of the vocal folds and are mostly characterized by the fundamental frequency. The filter is the result of the passage of the air by the vocal tract, responsible for the amplification of specific frequencies, the formants. Our results show a large contribution of frequency parameters originating in the source to individualization of the PURR. In turn, the frequency of the peak of energy, probably a filter resonance, apparently had a small contribution to the individualization of the PURR, as suggested by the lower peak value attributed to it. Another explanation for individual differences is related to the hypothesis proposed by Heisenberg, that PURR originated as a combination of cluck notes. Cluck is the contact call of the guinea pigs and informs receivers about the location and mood of the sender. Calls with these features are frequently associated with individual identity signals. We expected that dominance status was coded in the courtship call due to its importance in guinea pig social structure and male reproductive success. But since guinea pig courtship is a multimodal display, the dominance is likely to be encoded in another courtship signal, like the rumba or St. Mark. Considering isolated signals, our study is the first to investigate the individuality of the acoustic signal and its relationship to male quality. Bauchat demonstrated a clear relationship between the material of perineal gland and dominance. In stable social groups, higher ranks are related to the production of the material of the perineal gland and the frequency of gland drag. There is no study examining the relationship between the rumba and male quality. Also, there are no studies examining the relationship between the signals of different modalities used in the display. We believe that the different signals in guinea pig displays provide different information, as predicted by the multiple message hypothesis about multimodal communication, but further studies are necessary. Another possibility for the function of the PURR could be to attract the female attention to the rumba during courtship. The PURR also could be eavesdropped by other females and possible rival males. Based on the individual information transmitted by the vocalization, receivers can make an informed decision about the sender. In summary, We demonstrated individual differences in the guinea pig's courtship call, and it was not related to male dominance status. The first step to understand the female choice is to explain the function of the courtship signals. Further studies are necessary to understand the components of this complex display and how females responded to combination of signals from different modalities. This study is part of my PhD dissertation, which investigated guinea pig female choice and the features of male courtship display. The complete study was published on Animal Behavior some months ago. My PhD was funded by the São Paulo Research Foundation and CAPS. I would like to thank the Animal Behavior Society for the opportunity to present my work today, to the University of São Paulo, my lab back, and the co-authors of this work, Dr. Patrícia Monticelli, my supervisor, Dr. Fernando Frei, my co-supervisor, and Bruna Ferreira, my lab colleague and partner in bioacoustic analysis. Finally, I would like to dedicate this presentation to Brazilian graduate students and researchers who continue to resist and do science. Thank you.